Hello girls, before we start section 13.2 today, I want you to go through just a couple slides here that are just a review over 13.1 and just write the letters in your notes, and then we will take a look at these tomorrow and see how you did. So here's question 1. The role of a master plan in a building is similar to the role of which molecule? Question 2. A base that is present in RNA but not in DNA. Number 3. The nucleic acid responsible for bringing individual amino acids to the ribosome is Number 4. A region of a DNA molecule that indicates to an enzyme where to bind to make RNA is the Alright, now let's just take a quick review here of what we did in 13.1. All we came up with was a new word, transcription. And that we started with our DNA molecule. It uncoiled and divided, and we made a complementary mRNA strand instead of a complementary DNA strand. And the enzyme that was responsible for the uncoiling to make mRNA was RNA polymerase, which was different than the DNA polymerase that we used to make the complementary DNA. And then the other change was that our new strand did not have thymine in it. Every time we expected to put in thymine on an RNA molecule, we would put in a U for uracil instead. Now, quick practice here. I've given you a DNA strand, a short little chunk, and I want you to write what would be the complementary DNA, so the other side of the molecule, and then what would be the messenger RNA strand. So pause for a second, write those down. And now we'll check. So our complementary DNA should have been TAG, CT, ATG, and our messenger RNA have been very similar, except everywhere we had a T, we're just going to put in a U. So UAG, CUA, UG. And then just one more practice. And come up with the complementary DNA and the messenger RNA strand. And hopefully you came up with AGG, CAT, GC, with the messenger RNA being AGG, CAU, GC. Just one single base was different. So now we need to learn a new word, and the word is codon. And all a codon is a sequence of 3 bases. So in this picture, one of the codons is AUG. And a second codon is AAC. So anytime we have 3 bases together on our mRNA molecule, we call that a codon. So each codon is responsible for one amino acid. And there's a couple codons that are special, and the one that is probably the most important is the codon AUG. This represents the amino acid methionine, and it is what tells the process to begin, and we call it the start codon. So no decoding will happen until we get to an AUG start codon. And then there are 3 special stop codons. The molecule needs to know when to stop, when the protein molecule is complete, so it can be released to go into the cell and do its work. And we will see those in just a second. This is one chart that shows the codon combinations and the amino acids that they do code for. We do not have an unlimited number of codes or an unlimited number of amino acids, but the way this particular chart works is you start with your first letter, and that would be the letter in the middle. For example, if we were looking for AUG, we would start here with the A, and then we would go to the next ring and find the U, and the next ring we would find the G, and that tells us methionine. Goodness, excuse me. These charts come in various forms, but they all work basically the same way. And you just put your amino acids together to see what, you put your codons together, your bases to create a codon, and it will tell you which amino acid you are creating. The next step was processing the mRNA molecule. So it went through and it decoded the DNA, and what we needed to do in the cell, before it can do that, we are going to So our second step is called translation. We just went through transcription. We transcribed, wrote down the code, and now the code is leaving the nucleus, and we have to decode it. We have to figure out what in the heck that DNA wanted us to know. What information did it carry? And the whole point of this entire process is to create a protein. And a protein is often known as a polypeptide chain. We're going to see that a protein is made out of a bunch of tiny little molecules called amino acids, and these amino acids are hooked together by bonds that are called peptide bonds. So poly means many. We have many of these peptide bonds, and it makes a long chain. One way you can think of it is when you're a little kid, you had those little pop beads that would hook together. It's very, very similar to that process. This process is going to take place on the ribosomes. So you can see over here in the corner we have our ribosomal RNA, our ribosome just waiting to spring into action. And on this picture you can see we have our ribosome right here, our big blue molecule, and we have a new molecule. It's our tRNA. And then down here we have our messenger RNA. So on this screen we have all three types of our RNA getting ready to work together to translate this molecule. Well, what the tRNA does is it just is in the cytoplasm. The mRNA made in the nucleus, the rRNA is in the ribosome, and the tRNA is just in the cytoplasm. And what it's doing is it's waiting to be called in to do some translating. And on one end of the tRNA molecule is a set of 3 bases. And these are called anticodons. And they are going to be the opposite or the complement of the codon that is in our messenger RNA molecule. And you can see these two guys are getting ready to meet each other because our A's would pair up with U's. So what happens is the messenger RNA has gone to the ribosome, and the ribosome is going to read this messenger RNA molecule. And these tRNAs are going to come with their anticodons right here. You can see where it matched up. And this particular tRNA has lost its amino acid. It went right there, and it's hooking up with the next tRNA molecule. So every 3 sets of bases get read, and every time those bases get read, a new amino acid is brought in and hooked. And the tRNAs know exactly which one to bring. So this particular one, that was the anticodon UUU, knew to bring in the amino acid lysine, which is going to hook right here, and it's going to continue to make the chain. So you can see here, our chain is forming, we've got these amino acids being hooked together by their peptide bonds, and because we have a bunch of them, we now call it a polypeptide. When it's totally done, it's going to become a protein molecule. And this is going to continue. It's going to keep reading along in this direction, keep reading this messenger RNA chain until it gets to a stop codon, and then it will know I'm done working and I can release my polypeptide chain and my job is finished. This slide just sums everything up. We started with our DNA. We can tell this is DNA right here because of the thymine base. And every 3 bases, we can consider that to be a codon, whether it's DNA or messenger RNA. Our messenger RNA is the complement of our DNA, so C becomes G, G becomes C, T becomes A. If we look a little further down, we see an A becomes a U. This messenger RNA leaves, it gets decoded, and then it makes our protein molecule, and those amino acids are hooked together by peptide bonds, and the whole molecule itself we can call a protein or a polypeptide. On this page you can see another table that shows the different amino acids and how the codons decode them. Here you begin by finding your first position. So if we were looking for, let's say AUG, we would find our first position A, we would find our second position U, and then we would go over here and find a G that fits in with AUG, and AUG is here, and it's methionine. And it's very, very typical to see the amino acids abbreviated with just 3 letters, and that's fine. I'm never going to ask you to memorize the names of all of the amino acids. That would come in a much later, more advanced biology course. But you will need to be able to read through the table, and we will have some practice. One other thing to mention is you can see up here at the top were some of the stop codons, the 3 of them there, UAA, UAG, and UGA. If at any point the tRNA meets one of those stop codons, it knows that its job is done and it stops bringing the amino acids. So let's see if you can do the steps that we just went over. So I am going to give you the tRNA anticodons, and you will tell me the complementary messenger RNA codons. So here are the 3 tRNA anticodons, and we're just writing the complements. So you should be really good at this. Take just a second and pause the video. And then you can compare. UUU would be AAA, UGC becomes ACG, and AGA would be UCU. So we're basically doing something very similar to what we did when we were making the messenger RNA in the nucleus. We were going through transcription. So now I'm going to give you some messenger RNA codons, and I want you to tell me what would be the amino acid. So GUC. So take a look at the table. GUC would be which amino acid? UAC and AAA. So GUC becomes valine, UAC it becomes the amino acid TYR, and AAA would be lysine. One more to try. We're going to try CCG, UCG, and GCA. So you can pause, take a look at the table, and then check. to see if you were correct. Alright, this time I want you to tell me what codons could we possibly use if I gave you the amino acid. So we're just doing it backwards. So here's the 3 amino acids, and what you're going to need to do is find those amino acids on the table, and many times there's more than one, and write down what codons are responsible for bringing those particular amino acids. So pause the video and take a look. How'd you do? If it's confusing, please ask tomorrow. We will be working with this table a lot, and it's very important that you are able to easily find your way around it. Don't worry too much about this slide. We're going to try this site in class, just a real quick little simulation that takes you through transcription. Alright, and to wrap up, I have one more question to ask. A codon typically carries sufficient information to specify a or an, and then decide what it is. And be ready to talk about this in class tomorrow.